Hi, in this video we're going to be talking about developmental networks. To explain developmental networks, I'm going to be using an example of the spiky beetle. But before we get into the example, I wanted to talk about these symbols and what they mean. The green arrow means to activate, and the red T means to inhibit. Now to get on with the example. In this example, we have 5 genes in this developmental pathway which causes spikes in a spiny beetle. However, in the ectoderm of spiny beetles, not all cells have to have spikes. Some cells are smooth, and using this developmental pathway, we're going to explain how that can occur. In this hypothetical pathway, we have 5 different genes, and I used a different animal to signify each different gene. So for the S, we have the Siamese cat. For the P, we have the pig. The I, we have the iguana. The K, we have the kangaroo. And the E, we have the emperor penguin. And in this developmental pathway, this causes spikes. However, mutations can occur, and if a mutation occurs, we can have a variety of different results. We can either have spikes, or we can have no spikes. So we're going to go through 4 different mutations and see what effect they have. So in the first example, we're going to be removing the pig from this developmental pathway. Now when this occurs, the Siamese cat now no longer has the pig to activate, which means that nothing can activate the iguana, which means that the iguana can no longer inhibit the kangaroo, which means that the emperor penguin can no longer produce the spikes. So no spikes are made when the pig is removed from the developmental pathway. Next, we're going to remove the iguana. When the iguana is removed, the Siamese cat can still activate the pig, but the pig can no longer activate the iguana. And because the iguana is no longer activated, there is nothing to inhibit the kangaroo, which means that the emperor penguin cannot create the spikes. So there are no spikes when the iguana is removed. Next, we're going to remove the iguana and the emperor penguin. In this example, the Siamese cat can activate the pig, but the pig cannot activate the iguana, and nothing is there to inhibit the kangaroo, and nothing is here to activate the spikes. So no spikes are created when both of these are removed. And finally, we're going to remove the Siamese cat and the kangaroo. And when this happens, nothing is here to activate the pig. However, because there's no kangaroo here and it's inhibited, now the emperor penguin can create the spikes. So there are spikes when both the Siamese cat and the kangaroo are removed. So one reason why not all cells in the ectoderm have spikes, it might be because spikes are typically used for protections in beetles, and so only certain parts of the ectoderm, such as the top layer of the beetle, would need spikes, whereas the bottom layer probably, or the bottom part of the beetle probably would not need spikes. And so that's probably one of the reasons why not all cells in the ectoderm have spikes.